Bacterial plasmids are extrachromosomal. They are eliminated by treating with radiation. Transmission of different characters can be seen. Drug resistance transfer by bacteriophage involves transduction. Transferable resistance involves resistance to multiple drugs. Here plasmids play an important role. High degree of resistance can be seen here. Phage typing is useful as an epidemiological tool in Salmonella, Staphylococcus aureus and in Vibrio cholerae. Drug resistance is not transmitted by chromosomes. Chromosome does not transmit drug resistance. Plasmids, HFR and RTF, these all transmits drug resistance. Plasmids are involved in multi-drug resistance transfer. Plasmid imparts capsule formation. Plasmid imparts pili formation. Conjugation does not involve bacteriophages. Jumping gene is transposon. Microbial killing below MIC is known as post-antibiotic effect. Mechanism of direct transfer of free DNA is known as transformation. Transfer of some extra chromosomal material from one bacterium to other can occur via F factor, C factor, R factor. Endotoxin from gram-negative organism is lipopolysaccharide. The term viable not cultivable, that is VNC, is used for Mycobacterium leprae and also by Treponema pallidum. Brown's tube is used for steam sterilization. Prions are best killed by autoclave plus sodium hydroxide. Virus-mediated transfer of host DNA from one cell to another is known as transduction. Bacteria may acquire characteristics by taking up soluble DNA fragments across their cell wall from other species. Bacteria may acquire its characteristic also through bacteriophages as well as through conjugation. Lambda phage is lysogenic form that incorporates with host DNA and remains dormant. Biofilm formation is the phenomenon which is responsible for antibiotic resistance in bacteria due to slime production. Shine-Dalgarno sequence in bacterial mRNA is near AUG codon. Bacteriophage is a virus which invades bacteria. Bacteria contains both DNA and RNA. Horizontal transmission of R factor is by conjugation. Polymerase chain reaction was discovered by Carey B. Mullis. F plasmid of high frequency recombination is extrachromosomal. Asepsis means absence of pathogenic microbes. McConkey's agar medium is differential medium. Gamma radiations are used for sterilizing plastic syringes. The process of destroying all microbes including spores is called sterilization. Dry heat is the best method of sterilization of liquid paraffin. Blood agar is an example of enriched media. Conditions required for autoclave is 121 degrees Celsius temperature for 15 minutes. Sterilization of culture media containing serum is by inspisation. Sterilization of fiber optic bronchoscope is done by glutaraldehyde. Glassware is sterilized by hot air oven. The disposable plastic syringes are commonly sterilized by ethylene oxide. Selenite F broth is an enrichment media. In nutrient agar, concentration of agar is 2%. Blood agar is an example of indicator media and also enriched media. Endoscope tube is sterilized by glutaraldehyde. Robert Koch's assistant advised him to use agar instead of gelatin as culture media for cultivation of bacteria as gelatin melts at 27 degrees Celsius. Glutaraldehyde is sporicidal. Hypochlorites are virucidal. The operating temperature in an ethylene oxide sterilization during warm cycle is 49 to 63 degrees Celsius. The sterilization method for catgut suture is radiation. Decreasing order of resistance to sterilization is first prions, then bacterial spores, and then bacteria. Sterilizing agents include dry heat and also ethylene oxide. Chlorhexidine and isopropyl alcohol can be used for hand washing. Radiation can be used to sterilize bone graft, suture, and also artificial tissue graft. Spocidal agents are glutaraldehyde, ethylene oxide, and also formaldehyde. NaOH is used in the digestion and decontamination of sputum in smear preparation. The indicator used in autoclave is Geobacillus stearothermophilus. Prions are the most resistant to the action of antiseptics and disinfectants. Sputum disinfection is done by autoclaving, boiling, and also by cresol. Ethylene oxide, gamma radiation, autoclaving, glutaraldehyde, and hot air oven. These all are used for sterilization of surgical instruments. The disposable plastic syringes are best sterilized by ethylene oxide. Autoclaving is done in steam at 121 degrees Celsius for 15 minutes, 15 lb pressure. Culture media are sterilized by autoclaving. Glassware and syringes are sterilized by hot air oven. Lysogenic conversion is seen in Corynebacterium diphtheriae, Salmonella, and also in Clostridium botulinum. Crystal violet, methylene blue, safranin, and malachite green. These all are examples of basic dyes used to stain bacteria. Molecular technique used to study expression of gene is microarray using gene chip. Real-time PCR is also known as quantitative PCR. Here the risk of release of amplified product into environment is less. Real-time PCR uses fluorescent probe for detection of amplified product. Mycin is bactericidal antimicrobial. Mycin is effective against gram-positive bacteria and also it is effective against gram-negative bacteria. Oral typhoid vaccine is an example of live vaccine. External quality assurance, that is EQA. It ensures the reliability of future testing and also it ensures the credibility of the laboratory. External quality assurance establishes the inter-laboratory comparability. It also shows retrospective periodic assessment. Orthophthaldehyde is non-irritant. It is odorless. Nutrient broth is basal media. Prion is most resistant to antiseptics. A chest physician performs bronchoscopy in the procedure room of the outpatient department. To make the instrument safe for use in the next patient waiting outside, the most appropriate method to disinfect the endoscope is by 2% glutaraldehyde for 20 minutes. Heat labile instruments for use in surgical procedure can be best sterilized by ethylene oxide gas. The best way to prevent contamination of the PCR process is internal sterilizing agents such as uracil N-glycosyls. The best molecular typing method is PFGE. Paul Ehrlich is known for its contribution to microbiology in the field of theories on immunity. Peptidoglycan synthesis occur in 4 phases. Cross-linking of polypeptide chain occur in phase 4. Sedimentation coefficient of bacterial ribosome is 70S. Biocides are substances that kills all living microorganisms including spores. Chemical agents that destroy the microorganisms by interfering with membrane functions are phenol. In drug-resistant mechanism, there will be complete removal of target, which is the cause of resistance to vancomycin. Here, the most common mechanism is production of neutralizing substances. There is also an alteration of target in pneumococcal resistance. Deoxyribonucleotides, DNA polymerase or thermostable enzyme, magnesium or ssDNA or template DNA. These all are required in PCR. Penicillin is extracted from Penicillium chrysogenum. Flagella is made of smaller units called as flagellin. Bacteria with polar flagella move very fast. All flagella essentially have same chemical composition. Endoscopes are disinfected by manual cleaning, disinfecting and by rinsing. In a postoperative care unit, 5 patients developed postoperative wound infection on the same wound. The best method to prevent cross-infection occurring in other patients in the same ward is to practice proper hand washing. Next question. Endoscopes have been known to be contaminated with Mycobacterium chelone, which is due to the poor quality of rinse water. Structure that is found in gram-negative bacteria but which is not found in gram-positive bacteria is outer membrane. Exotoxins are highly antigenic. A soldier developed thigh abscess following gunshot injuries during an anti-insurgency campaign operation. Gram stain of the foul-smelling exudates revealed plenty of pus cells and a mixed flora of gram-positive and gram-negative rods. However, aerobic culture was sterile. Which of the following enzyme is most likely involved in the following reaction? The reaction is 2O2 plus 2H plus equals H2O2 plus O2. So the following enzyme here is superoxide dismutase. On gas pack, 2 types of bacteria was isolated. Biochemical studies revealed that these bacteria failed to produce superoxide dismutase, catalase and peroxidase because they have enzymes only for the EMP pathway. Glutaraldehyde is poricidal, hypochlorites are virucidal, ethylene glycol is intermediate disinfectant. Bacillus stearothermophilus is used for biological monitoring of gas plasma sterilizer. All microorganisms are best defined as organisms that are too small to be seen with the unaided eye. Endotoxins are found only in gram-negative bacteria. B subunit of the exotoxin is the binding portion of the molecule. Most gram-negative bacteria have a lipopolysaccharide which is found in the outer membrane. Probiotics exhibit all but the following characteristics remain viable for a long time. Timing of blood culture collection need not coincide with fever spikes. Phenol is bactericidal at a concentration of 1%. The most ideal disinfectant for preparation of skin prior to insertion of a central venous catheter is 2% chlorhexidine. Bovie-Dick test is used to assess the performance of steam sterilizer. The spore of Geobacillus stearothermophilus is used as biological indicator to monitor steam sterilizer. Universal or standard precaution should be applied to blood, body fluids and also for sputum. The first thing to do in case of accidental occupational exposure to blood and body fluid is to wash with plenty of soap and running water. Vaccines are sterilized by filtration. Disease transmitted by blood transfusion are syphilis, malaria, AIDS. Microorganism used as weapon in biological terrorism is smallpox virus. Bacterial spores are resistant to boiling. Bacterial spores contain multiple layers. Bacterial spores are formed under adverse environmental conditions. The chemical nature of capsule may be polysaccharide or polypeptide. Most important surface active disinfectants are cationic. The purpose of staining cells on a microscope slide is to add contrast in order to see them better. A microbiologist inoculates Staphylococcus epidermidis and Escherichia coli into a culture medium. Following incubation, only E. coli grows in culture. Here, the culture medium must be selective. You are observing a cell through a microscope and note it has no apparent nucleus. You conclude that it most likely has a peptidoglycan cell wall. Coates postulates are criteria used to establish that a specific microbe is the cause of a specific disease.